I'm now delighted to introduce you to our next speaker for this event, featuring Dr. Peter Nag. Dr. Nag is board certified in surgical and molecular genetic pathology with training at Stanford and leadership roles at the University of Iowa and Columbia University. He was the first to introduce whole exome and transcriptome testing for constitutional and somatic disorders. Dr. Nag will be teaching us today about accelerating prenatal diagnostics, faster answers for expecting parents.
My name is Peter Nagy. I'm Chief Medical Officer of Praxis Genomics, and I'm going to talk to you about how to accelerate prenatal diagnostics. That is, provide faster answers to expecting parents. Having a child is one of the most exciting and wonderful things a couple can experience. And myself, I have 3 children. I went through this 3 times, and every time the main concern is the health of the child to be born. And currently, there are pretty good non-invasive combined first trimester screening tests to detect common abnormalities such as trisomies or severe heart disorders or spina bifida and so on and so forth. So the main tests that are used currently are blood tests where these measurements are done from the mother's blood, and the fetal ultrasound where they actually visualize the fetus and can notice gross abnormalities in size, in development, and they can make deductions about the conditions of the heart and the various organs based on what they see. There's another test that was introduced in the last decade or so, which is non-invasive prenatal testing. In this case, again, they take blood from the mother, but instead of actually looking at changes in the mother's blood, necessarily they're looking for pieces of DNA originating from the fetal placenta in the mother's blood. So they are basically trying to do genetic testing on the fetus using the actual placenta as a surrogate, really. And they can evaluate with this methodology whether there is a normal number of chromosomes or there is some suggestion of trisomy 13, 18, 21, or maybe the sex chromosome numbers are abnormal. There is also a big push to extend this sort of prediction to other regions of the genome, which are getting smaller and smaller in size, increasing the scope of this test. However, this increased scope also increases the false positive and false negative cause, simply because, again, you are not really testing the embryo, you're testing the placenta, and the amount of fetal blood or fetal DNA in the mother's blood is quite low. So combined first trimester screening that I mentioned to you, which is just measuring certain proteins and substances in the mother's blood, has a false positive rate about 3 to 5%. The NIPT test, which I mentioned to you, which actually looks for pieces of DNA of the placenta in the mother's blood, the false positive rate of this test is 0.13%. But this is only true when the test exclusively targets chromosomes 21, 18, and 13 to detect trisomy. The false positive rate increases when testing for abnormalities across the genome. And because of this, let's say out of every 1,000 sample, every 1,000 pregnancy, there is one, let's say, that will require confirmatory testing. Confirmatory testing is done by invasive procedures. There are two ways. Either they take chorionic villus biopsy that can be done earlier, or they do an amniotic fluid sampling, which can be done later and provides or poses a smaller risk to the fetus. The risk for abortion is below 0.5% or so, but there is definitely a risk. But in the case when there is an indication for invasive testing, such as the ultrasound shows that the baby's neck is swollen. We call this nuchal translucency increased, between 3.4 and 9.6 millimeter, or there is a general swelling of the baby, which refers to as hydrops, usually indicating a heart abnormality, or there is brain abnormality, nose bone is missing. These are sort of indications for various problems. NIPT results, where they try to look for mutations in the fetal DNA from the mother's blood, can be inconclusive, let's say because the mother has autoimmune disorders, or it could be, for example, reporting out that the sex of the fetus is not matching because the pregnancy started with a twin pregnancy and one fetus died, and the DNA from that fetus placenta still hangs around. And, commonly people also want to do this test when the mother is carrying a fetus from the husband's sperm and the donor egg. Spina bifida is a very common indication when you see that the spinal column didn't close properly. You need to exclude in these cases, you need to exclude that it's part of a syndrome, because clinics who correct these kind of problems, they do not perform the surgery if it's part of a larger syndrome with additional problems. So what do we offer to help this invasive testing, which usually takes about 2 months to get the results from, when it comes to the genetic analysis, of the cells that were removed from the chorionic villi or the amniotic fluid. So what we do is we perform whole genome sequencing, which doesn't require culturing. It doesn't require maternal cell contamination testing separate from our sequencing, because our programs can detect maternal cell contamination right away. So basically what the traditional clinics, laboratories can provide you in like 2 months, we can provide you the same answers in like a week, or so and you can go in an order of how extensive you want the testing to be, because the data that we obtain is from the whole genome. So we can immediately upgrade if we do not find an answer that basically solves the question posed by the clinicians. So we start out with analyzing the genome sequence for aneuploidy, basically testing how many chromosomes there are in the child. And this is sort of a microarray equivalent. But we can then go on and look for point mutations in the coding region of the genes or in the whole genome. And this can be done immediately. We can also do additional testing, which requires cultured cells. And these tests are for specific disorders that cannot be diagnosed with short-read sequencing. And also for transcriptome sequencing that we use to immediately provide you with the functional significance of a mutation if it changes the processing of the gene expression. So the 1 week turnaround time is great because we can provide you answers that you can act. And oftentimes we get these requests in time of holidays and right before 24 weeks and so on. But you need to have your samples also cultured, because there would be a need in some cases for follow-up testing with classical cytogenetics, or optical genome mapping needs to be done to detect translocations. So in summary, NIPT testing and fetal ultrasound results must be confirmed, and any inconclusive results must be evaluated using genetic analysis of amniotic fluid and chorionic villi. Direct whole genome sequencing of these samples allows for a definitive genetic assessment in 1 week. Long-read sequencing and optical genome mapping allows for excising of expanded repeats or facioscapulohumeral dystrophy diagnosis, which are special disorders that can be inherited through the family. Transcriptome analysis provides a functional assessment of the findings, but don't forget, culturing cells from the samples is still recommended, since detection of mosaicism below 20% is not guaranteed, and only classical cytogenetics can do that sort of assessment. So thank you very much for your attention. And if you need any additional information, please go to our website, www.praxisgenomics.com. Thank you.
Thank you, Dr. Nag, for